So today we're going to continue talking about protein characterization, and in particular about how we can detect and analyze what other proteins a given protein interacts with. And the basic idea is that most proteins function in complexes where they interact either tightly or with each other or loosely with each other. And it's especially, the most straightforward is when they interact really tightly with each other and they, the binding strength is high between them. But the idea is that if we have a protein complex, we can either tag genomically one of the proteins or have a very good antibody against it. And then, of course, this protein complex is in a mixture of proteins naturally in the cell. But then when we try to pull out the complex, it's going to be exposed to proteins that it doesn't usually interact with, that it usually doesn't see at all in vivo. So that's going to introduce a lot of sort of spurious connections. So if we often see, for example, that when we do IP, that very high abundant proteins in the cell could come along easily. So we usually use magnetic beads with an antibody against the tag or directly against the protein, and then we try to wash away as many of the proteins as possible, the non-interactors, the non-specific interactors. But the problem is that when we wash, we usually remove some of the proteins that are not very strongly associated with the complex. So we end up with a case where, in general, where we get just a part of the protein complex and we have non-specific interactors also. So that's a lot of what we're going to talk about today, is about how one can keep as many of the parts of the protein complex and remove the non-specific interactors, either through different experimental methods or with computational methods. So then now, when we have this, we measure it with mass spectrometry. So we go through the regular workflow where we digest the proteins, analyze the mass spectrometry, and identify which proteins are there. So people have worked quite a bit on how to optimize the affinity, and it makes a big difference if one does this properly. And this paper actually is some of my collaborators. It came out yesterday. Sorry?
Quite recent.
Yes, quite recent. But it's been, I've used this slide for a long time. It's been in the works for years. But yeah, the paper is quite recent. So the idea there is that every protein complex is an individual, in the sense that it needs specific conditions. So you need to optimize the salt, the buffers, the detergents for every specific protein complex. So this is this technology that I'm going to talk about now was developed at Rockefeller University, and it's the steps are to that one, before doing the affinity, one has the frozen cells, and one uses this grinder which mechanically breaks down the cells into small pieces. And ideally one wants to break them down into pieces that are of the order of magnitude of the protein complex that one studies. But that's very difficult, and usually it's still quite a lot bigger. But that's the idea. And the way this works is that you have these metal balls, and you cool this in liquid nitrogen, and just by the bowl and move this whole jar so that the balls crush the cells. And so then the next step, one has the grind date, and that's what this paper is about, is that an robotic system that tests many different conditions. So it uses a 96-well format, and you can program it to try different detergents, buffers, and salt concentrations, and different kinds of salts. Then it's automated, so you have the filtration step here, and then finally the readout that we usually use is the STS page gel. And the reason for that is that it's highly parallel, and if you want to screen a lot, it takes a lot of time to do mass spectrometry. So the results look like this for one complex. So here we can see by varying the conditions, we can go from conditions where we only see a few proteins here that are associated with our bait, to cases where we see slightly more proteins, like these two here are one sees under these conditions, and then two cases when there is mainly background and it's not a good condition at all. But the idea is that by looking through this space, we get very different pullouts. So going from just probably the tightest bound proteins to we can add on layers and layers, so that's the idea. And the other thing is that here, what this one of the figures from the paper shows, that there are a few complexes here, and as you see, the optimal condition for these complexes is quite different when it comes to both the buffers, salts, and detergents. So it's definitely worth exploring the space of conditions, and it's usually manually. It's quite tiring to do that. So that's why we try to develop and automate this.
Is there a property of the protein that will suggest this is better to use?
So we don't have enough data, but we've also set up a database where we're storing all this information, and actually we're storing the actual gel images to be able to go back when we collect enough information to try to then determine if there is some way to make sense of it, and then of course if one has a new protein, if we can predict.
Yeah.
But we really don't have enough data for that yet. So now one can also look at protein complexes inside the mass spectrometer and measure them in there. So this is from several people have done this. I mean, there are a few groups that do it really well. So the idea is that one affinity purifies a complex of proteins, and then the first step is the same as one always does, that one just identifies the components, and then one can go back and by knowing what the component list is, one can then measure the intact mass of the complex in mass spectrometry. And then usually these complexes are very highly charged, and but we can measure the mass very accurately, and if we know the component list well, then we can determine how many, what's the stoichiometry of each of the proteins. So that's, but then we can also, in the gas phase, fragment the complex and measure the fragments. And what usually happens when we fragment a larger complex is that it falls apart asymmetrically, so that there will be most of the proteins will stay, and a few will or a small complex will fall off, and the charge will also be distributed in a way that the large complex will keep much less of the charge. So that's why even if we have an ion trap or some kind of trapping device, it's difficult to go back and continuously fragment, because the most of the charge stays with the small, smaller part. So the third thing we can do is to partially denature the complex in the, sorry, this is very small here. But denature the complex in solution before we do mass spectrometry. And there we actually have quite a lot more different options than what we have when we do the fragmentation in mass spectrometry, at least so far. And then the idea here is to put this all together and build some kind of interaction network. So at least which one interacts with which. And these are, of course, not that detailed models, but it's still, one can find subcomplexes, and one can see by combining all this information which ones are closer to each other in the complex. So one can start building up how these complexes are built. And so this is just one example here where we're looking at it's the from yeast, it's the APC subunit, and we can determine the stoichiometry this way, but not for all of them. Not APC9 we couldn't. And here we're used to very exact molecular weights, but now when we measure these very high molecular weight complexes, we have quite much larger uncertainty. And that's the reason is that you see that these peaks are, there's a lot of peaks that are not very well resolved, and we have often adducts. So that's what makes it a little bit more difficult. But then when we have fragmented, we see that we have much better resolution, of course, when we get down to single subunits of proteins. So now one example of where this kind of information, in addition to a lot of other types of information, was used to make a structure was this work comes out of Rockefeller University, also from Mike Rout and Brian Child's labs, where they collected all sorts of different information, and then from that use these different kinds of information as constraints to make a structure. And you see that this is not a atomic resolution structure, of course, but it says tells you where the different proteins are in the. So this is for the nuclear pore complex in yeast, and you can see how nicely how the different rings are organized, which different proteins are there. So the kinds of information they used is they did ultracentrifugation. They did quantitative immunoblotting to get the relative abundances of the different proteins, affinity purifications to get subcomplexes, then other things like electron microscopy to get shapes, and then mainly was a big bioinformatics component to put all these constraints together. And also that there you have to have, sometimes there are, of course, constraints that are contradictory. So that you have to sort out, and then you have to put different weights on different kinds of constraints that come from different experiments. So if we look at affinity purification, there were, for each of the components, or for most of the components at least, they did an affinity purification to see what the different, what the highest affinity interactors were for those. So this just shows for each of the knobs that were pulled out what other, and this took a lot of time, and it was done over probably 10 years or so. And nowadays it could be done much faster. So another example from Eliana Krista's lab at Princeton is where she looked at histone deacetylases and looked at their interactors. And you probably recognize this paper that we discussed recently. But this just shows another example of what people have done, and there are many examples. This is probably what mass spectrometry-based proteomics has been most successful at, and most people are working on, is to elucidate components of different protein complexes. So then, as I mentioned, the question is, how do we distinguish between specific and non-specific interactors? And this slide shows one method to do that. And the idea here is that you do lots of different, you have a lot of different baits, so you do a lot of different experiments, and then you look at, use the whole information to estimate whether, to tell whether something looks like more like a non-specific interactor or a specific interactor. So the idea is that non-specific interactors will have a quantity that's much more similar over many different baits, and the specific ones will be specific only in a few of the IPs. So that's so this is based on also on spectrum count, as we discussed before. That the one way to quantify is to count the number of spectra that we can use to identify peptides that belong to a protein. So that's reasonably good way to quantify, especially if the spectrum counts are not very low. But the idea, so this is one method that where they look for outliers, and and here also what they did is to they do simulations to get the random distribution of what it would, to determine what would be a significant value, what would be a significant outlier. A quite, I would say, conceptually quite similar method is what's probably, I mean, nowadays more used is SAINT, which also is based on that you you have a lot of several experiments, and that you can do this comparison over within an experiment and over many experiments. And one thing that they in SAINT you can either have a negative control, or you can also do it without a negative control. And this the modeling here is based on the same way as what was developed for identification earlier. So the idea is that there is a distribution of false interactor as and a distribution of true interactors, and you set, yeah.
So from what I understand, non-specific interactor would be same for all IPs. But what if this non-specific interactor is actually an interactor for a for a IP? Like, I mean, it's because it's, let's say, it's abandoned its IP with all the IPs, but for, let's say, one IP, it is actually an interactor.
Yeah. So that that's fine in these methods as long as you get there is a higher quantity. So you have as long as you have a higher spectrum count. So you can even if you have a low level over many of your IPs, and then but in one it's higher, that's it will be okay. So that, yeah, so it can detect that. And now, so they have also taken a step further, which they made a database of interactors that people find over many IPs. So they call it the crapome. It's and that's because the idea, I mean, the problem is that most people don't do many IPs. You have one protein that you're interested in, so you do an IP for that. So the nice thing with this database is that then you can use that. Use everyone else's experiments as your comparison, and that's also has been pretty useful for a lot of people, and people like the name very much also. So one way that you can do this that we've mentioned before is you can also experimentally look at, try to distinguish the specific and non-specific. And one way of doing it is to use heavy isotopes. So in this case, we are growing our strain with a tag protein in light media, and we're growing the wild type in heavy media. And then we mix them together and do the immunoisolation together. And then when we do mass spectrometry, the ones that are specific we see as only the light peptide, and the non-specific ones we see both heavy and light, roughly the same amount. So this works very well. So there's one main issue with this is that if you have fast exchanging proteins that while during the time of the immunoisolation will go in and out of the protein complex, those will also show up as non-specific. So this is quite a conservative method, so that the ones that you show up as specific, they are specific, but you will have false negatives, because several of the specific interactors will show up as non-specific because if they're fast exchangers. So then another method that people have developed is to do chemical cross-linking. So we had the problem that when we wash our beads to get rid of the non-specific interactors, we also get rid of specific ones. But if we have cross-linked our complex, then we can wash much harsher and still keep. And if we only interested in looking at interaction partners, we actually don't need to do much cross-linking. We can just do a few over a protein complex, a few cross-links to stabilize it a little bit. And that's so the idea here is that we have it cross-linked, we can wash it much harsher. Then we do the regular thing, we digest, we analyze the peptides by mass spectrometry, and then we fragment it to get MSMS spectra to identify now the components. And again, as you see, we don't, in this case, we don't even try to find the cross-linked peptides, we just use the cross-linking to stabilize. But of course, so here we can, since we're not interested in the exactly the peptides which are cross-linked, we can use something like formaldehyde that can cross-link quite a lot of different amino acids together. So, but we do it just very lightly. And the idea here is that one can either cross-link in vivo, and then we have to be very careful, of course, with the cross-linking, because that will really poison the cells and change a lot of things. But the idea in this slide is that one can take the cell grindate and do the cross-linking there. And then we don't change the growth conditions of the cells, but we still get a very good stabilization. So this is just one example from nuclear pore complex again. So the black are the when we don't do any cross-linking. So the tag here is on NAP84, which is part of the outer ring. So you see when we don't have cross-linking, we see mainly other outer ring proteins that correspond to these black. But if we do formaldehyde cross-linking, which is shown in red, we pull out almost the whole nuclear pore complex in one go. And we also get it becomes much cleaner. So this is something that people are experimenting more and more with. I mean, actually, people have been working with cross-linking for many, many years, but it's really only recently that several labs developed methods to make it very more generally applicable. So, and especially what people are interested in is finding the actual interaction sites. So to figure out how either to use it to determine the structure of one protein or find the interaction surface between two proteins. And of course, this is a much more difficult problem because here we actually have to find peptides that are cross-linked and peptides that are cross-linked to between amino acids that are far away in the linear chain, otherwise it doesn't give much information, or that are between different proteins. And so the other problem here is that there are lots of possibilities. So if we have we do the trypsin digestion and we have a few different possibilities. So usually now people use more specific cross-linkers than formaldehyde. Things like that only react with arginines, for example, or that. So there are lots of different ways that these can be combined, and there. So if we have n peptides with reactive groups, it's roughly n square possibilities of the order of n square possibilities. So for example, if we look at protein A and IgG heavy chain, there are 990 possible peptide pairs. But if we look at the yeast nucleopore complex, there are a million possible peptide pairs. So that's, and on top of this, these are just a pair. Then there are lots of uninformative forms, like where we just get a single cross-linker as a modification, or then modifications that are, cross-linking that happens between amino acids that are very close to each other in the linear chain, and so on. And since we have all these different possibilities, the signal gets diluted, and we also have the problem that mass spectrometry we have a limited dynamic range. Those two in combination makes it quite difficult. So what we want to do is preferably try to enrich for the cross-linked forms. And that's what one of the recent improvements that came out a few years ago is that by using size exclusion chromatography, one can enrich for the cross-linked forms. Then the other problem is that, of course, they both peptides that are cross-linked have to be long enough so that we can identify them. Then, because we usually, when we work with tryptic peptides, we can fragment them well, but now we have two tryptic peptides together, so the mass will be higher. And that's why often people have better success with electron transfer dissociation that works better for larger peptides than collision activated dissociation. So then, so one big thing that we need to study protein-protein interaction is good antibodies. Either good antibodies against a few tags like protein A and green fluorescent protein, or a few, or against the antibodies themselves. The proteins themselves. So one thing that one wants to be able to do is then to generate antibodies easily. And one way to generate antibodies is if we can sequence them. So now we also know that antibodies, if we have know the genes, antibody genes, they go through main steps of recombination. First is the VDJ recombination, where different genes are combined. 3 different genes are combined together to make up the antibody. In this case, looking at the heavy chain, and it's similar for the light chain. And just to take all the combinations of this would create a very large database. But then on top of that, there is a second step with the somatic hypermutation, which will introduce mutations into the sequence. So we really can't, like in most, when we study most proteins, we're pretty okay with just using the reference database. But here we really need to make a specific database for each sample and see what antibodies actually the body produces. So one way to do this is, in this case, this is an example of where and this was someone who was infected with HIV, but did not get AIDS. So the immune system mounted an appropriate response and was able to hold the HIV at bay for many years. So this is really, and without any drugs. So the idea was that if this person makes good antibodies, we should try to figure out what kind of antibodies these people make, and see if one can maybe then make a vaccine that so other people would generate the same kinds of antibodies. And so there were 2 steps to this. So first, B cells, high affinity B cells were selected, memory B cells were selected, and then sequenced to make a database now of the antibody repertoire of this individual. And then the other step is to take the serum and select the high affinity circulating antibodies and measure those by mass spectrometry. And then together with this, we can search the mass spectra in our database. And this just shows one clone. So you can see that there's quite a big clonal expansion with variations in the sequences that we have in this one clone. So now, but if we want to make reagents, it's the problem with human antibodies and with most animal antibodies is that they have a heavy and a light chain, and the specificity is made up of a combination of the heavy and the light. And it would be nicer to have just one amino acid sequence that one could easier work with. And the nice thing there is that llamas actually have a single chain antibody. So camels and llamas both have that. And so the way it works, so they have just a single heavy chain. And then this is ideal for mass spectrometry, because if we can determine the sequence of this region, we can then make it very easily, make a plasmid and produce it in E. coli. So we have a monoclonal antibody then. And it's small, so that we can easily work with it. We can maybe change it it folds well usually, and it's very cheap to make. So the question is, how do we then sequence it? So we developed a method to do this, and the idea is that one gives the antigen to the llama, and then after it has incubated for a while, takes a bone marrow sample, and then does targeted sequencing of the single chain variable region. And then that will be our sequence library that then we can search. And then from the blood, we fractionate and select the fraction with the single chain antibodies, and then do sequencing with mass spectrometry. And then we have our library that we can then translate after cleaning it up a little bit to get the peptide sequence library that we can then search with a standard search engine like Xtandem. Oops. And sorry, the colors on the are better on the screen. And then we have our identify peptides that then we can then map back to the primary sequences to select out which sequences we should express. So if we look at the sequence library a little bit closer, so this was sequenced with MySeq, so we have about 300 base pair paired sequences, and we have a big overlap that's about 200 base pairs in this case, because the region, the variable region is small. So the quality wise, like usually the quality drops at the end, and so we trim off a little bit at each end, but the overlap is quite big. And the nice thing is that where the quality of read 2 is low, which is over here, the quality in read 1 is still pretty high. So it's easy to combine them together. And after merging, the quality is very high of the merged reads. And we see that we get a distribution of reads, and the peaks are 3 nucleotides, has a 3 nucleotide separation corresponding to, of course, 1 amino acid. So now we have a database, and we can search it. And these are just a few examples of spectra that match to our peptides in the database. Then now we can take this and score the different sequences. So we want to rank all the nanobodies in our database to select out which ones we should try to express, which one seems to be high affinity binders. So the main sort of things that go into the score. One is, what's the coverage? What fraction of the whole sequence do we observe peptides from? Then looking at what coverage do we have in the CDR regions, and because those are the most variable parts of the antibody, and especially the CDR3. And then looking at other parts like the scaffold regions, and finally how many counts do we get in the sequencing. And then we group them together, and finally we end up with a list of ranked nanobodies. And this is again a little bit difficult to see, but then here is the sequence of the nanobody. And then underneath are the different peptides that were identified. So now when we have a sequence, we can then synthesize the gene and codon optimize it for E. coli, make an expression vector, express it, and then see how we can simply purify it and see what yield we get. And then test it, how well it works for this. So the first that we tried this on was antigen that we tried this on was GFP. So this now lends itself to easily test by either looking at the fluorescence from the GFP itself or from a fluorescent tag that's attached to the nanobody. And as you can see here, we get a very similar picture. So now the other thing that is nice with this method is that you don't get only one antibody. You get several. So in the case of GFP, we got 26 different antibodies. And then we used NMR to see where on GFP they bound. So this shows one clone that it binds where the white atoms are. And then there was a second one that bound on the other side. And now you can probably see that what we can do is that if we do the overlay, they don't really interfere with each other's binding regions. So what one can do is to make, by taking two reasonably good antibodies that have low KDs, but if we then add a peptide linker, together the two will have really a very high affinity. So one can now play around with this and use this for different purposes. And it's relatively easy to do this whole process. So one example of just a looking at interactors for a virus, so this is HIV, and then, as you know, the HIV genome is extremely compact, so everything is seems to be very optimized in it. So it's in this case very difficult actually to put in a genomic tag anywhere without disturbing the virus and making it less viable. So in this project, the interactors for the envelope protein and for WIF were looked at. So if we then look at envelope, this was also a collaboration with Brian Chait's and Mike Rout's labs at Rockefeller, and also Mark Musling at Aaron Diamond. So they worked out a method to do, or they found a method to do a random insertion of 5 amino acids. So this now shows the envelope gene. So it has 870 nucleotides. And so after random insertion and sequencing, show that we get random insertions all over the gene, but not at all. This is a log scale, so it's not at all even the insertion, but we still get quite a lot of diversity. So then the next step was to then let these different strains compete with each other what happened. So these are three experiments, and then we see at day 1 already there is much fewer. A few of the, I mean, a lot of them fall out immediately, depending on where the insertion is. But then we see after a few days, there are really only 2 positions where insertion is tolerated. So in all these 3, it ends up being the same.
If they compete with the wild type, are they fair well?
Yes. So wild type is also so the wild type is part of the competition, and these fare well. These 2 fare well also with wild type. But this is a very nice thing because we can actually measure the fitness landscape of this gene, or where, I mean, depending on where looking at insertions, and we can also look at evolution. So now again, in this project we used the heavy isotope labeling to sort out the specific and non-specific. Sorry, this was in the wrong order. This was the result for envelope. So now it was done, so either it was done both so that the tagged was in light medium and the wild type in heavy, and then we switched it also. What we see here is that there is a bunch of proteins up here close to the 1 for eye-to-eye ratio. So these are the really specific ones. And then we have a lot of proteins down here at 0.5, which are the non-specific ones. So you can see if we go along here, you can see the histogram. So there's a peak at around 1, and then a peak around 0.5. Then there are actually a whole bunch of proteins here in the middle. And these are the ones that are fast exchangers. So that's why sorry, again, you can't see that probably on the screen very well. But there are a whole bunch of dots here also. And you can see this background in the middle. So that's so this seems to work really well for one can find many interactors this way. So now I'm going to move on to microscopes. So far we talked about how with mass spectrometry we can identify interaction partners. But we really also want to go a step further and see how the details of these interactions. And then and microscopy is, of course, very powerful at giving us the spatial resolution, localization, and so on. But we have this limitation that depending on the wavelength, we can't really see objects that are resolve objects that are smaller than the wavelength. But what people observed was that if you have an isolated object and it doesn't move and you have long enough time, you can actually measure even if the bit of the distribution, so this is now 250 nanometers, you can measure the centroid of this very accurately, in this case down to 1 nanometer almost. And this is the idea that with super resolution microscopy, what we can do is we can go from looking at the width of the distribution to instead replace that with the centroid. So now the problem, of course, is that we don't have isolated objects in cells, but the proteins are close together. So if we look at this with light microscopy, we would have just a big smear. So now the idea is that if we can't isolate these in space, what we can do is we can separate them in time. So we make these blink at random, but at a very low frequency. There's a very low probability that each time point a molecule will blink. So so what we'll have is that we can separate them and then replace each of them with the centroid that we measure. So this way we can actually so this is in collaboration with Eli Rothenberg in the biochemistry department, and he has worked out this method so he can get about 20 nanometer resolution this way. So there are several different versions of this. So that were developed about the same time, a little bit less than 10 years ago, and it was awarded the Nobel Prize in Chemistry last year. The super resolution look as a. So one is PALM, where you have a fluorescent protein itself, or STORM, where you have an antibody that's labeled. So now, if we to give you one example of how one can look at interaction partner this way, is from looking at the intercalated disk. So here, the intercalated disk has a few different junctions. There's a tight junction, the desmosome, and the gap junction. And these were believed to be quite separate entities. But our collaborators, so this is with Eli Rothenberg and Mario Delmar, there was some evidence that plakophilin 2 from the desmosome interacted with connexin 43 from the gap junction. So what they decided to do is to look at this more in detail. So now this is a regular microscopy image of the gap junction of the intercalated disk, and the intercalated disk is here and about here. So these are the cells. So for example, one cardiac cell is here. So now if we turn this into super resolution, we get a much better. So that's what's the difference between regular fluorescent microscopy and super resolution. And so if we look at it closer, we can see really what that sometimes. So this one is close but doesn't interact, but others are interacting. So for example, another example, more detailed, which is regular microscopy and super resolution microscopy. So then now this gives us a much more detailed view, and we can really tell a lot about co-localization. So I mean, there's a very big difference between this type of co-localization, where people are in the same space but don't interact, or more meaningful co-localization. So that's the sort of difference that we can do with super resolution. So then now it's relatively straightforward to, when we have these images, to extract all sorts of statistics. So for example, we can get distributions of areas, distribution of circularity, for these different molecules. So one observation that they made was that the connexin 43 area seemed to be correlated with the co-localization area. So there was some background with low co-localization, but this gray area between 100% overlap and 50% overlap was much more populated than the area below the 50% overlap. Now, this looked very promising, but one thing that we wanted to test is, of course, is this just random, or do we actually, is this really significant? So, and the other observation they made is that if anchorin G is silenced, then the areas increase. There's a loss of circularity for connexin 43, and this correlation between area and colocalization is much less pronounced. So what we decided to do was to do Monte Carlo simulations. So to take all the measurements from the experiment regarding density of molecule size and shape, and then randomly put them down and see if one would get a similar co-localization as in the experiment. And so what we do here is that we take the experimental measurements and then randomly sample from these, both for connexin 43 and plakophilin 2. And this just shows that this is the experimental distributions, and then in the simulation we can successfully sample from these same distributions. So then the question then, was the observed overlap random, or was there, did we measure something? So in this case, this is our random point, and this is what the experiment. So now there's no, so this axis here is attraction between the two molecules. So what we get here, and the nice thing, in addition to just being able, we also from the simulation can estimate the error bars in the measurement. So what we see here is that both in the for both areas, we see that a random model does not explain what we see. But then we can introduce an attraction between the two molecules, and then we see that we can get within the error bars of the experiment, both in the untreated and anchorin G silence case, but the attraction factor, it's difficult to see here, it's a bit small, but is much lower in the anchorin G silencing case. So as we suspected, the anchorin G silencing is becoming more disordered, but still not completely random. So that was about what I had to say about protein interactions.